Jean-Baptiste?
Oui. Je t'entends. So good afternoon, everyone. Thanks so much to having you here for this second edition of the Microbial Sensors for Water. So we have an interesting afternoon of diverse talks with 2 sessions that we will start very soon. I will give you first a very brief introduction and also an update on the community of practice that has been established during the last event. So let me start. So again, here's the program: 2 sessions, coffee break in between, and a QA session at the end. So all started actually in December 23. We had a first event edition of the MicrobialSensors4Water. It was a technical day organized around 4 sessions. It was here in Belleval in Luxembourg at a list, and also sponsored by, you can see below, different companies. And it was a really successful event. We were about 70, 80 people on site with very interesting presentations and really rich discussions among the community. Different nationalities, but also researchers, utilities companies, governmental agencies. So the objective was really to bring together people around this common thematic and share our knowledge, our experience, and also questions about these new technologies. At the end of this technical day, we wanted we did an online survey with the attendees to question the interest for potential community of practice that could be set up, and so to have something continued and keep in touch among the community. The idea would be to bridge between also promising RTD outcomes and marketable products, exchange among users, and the community here, and also foster the development and uptake of these new tools to maximize public health benefits. So the survey was really conclusive, so it was really a big interest of the community to set up such a community of practice, which we decided to do. It took a bit of time with Corina Carpentier from Sensilo, who was setting this up. And the website, so the platform, the online platform, which aims to be participatory, is being hosted by Sensilo, and it is now online, but not yet completely finalized. So in a couple of weeks or months, we will be notified once it is done, and you will be able also to download the contents and also participate to this. Because the idea here is to make available the slides from the different MS4Water editions, of course, the last one and this one, but also to announce new papers around online monitoring, new conferences, also potentially white papers, reports, and so on that are publicly available. That to actually bring them here on a platform so that it is centralized for everyone. But also we were thinking about having a discussion forum for users, so we can also share directly among users. So that's still to be put online, but you will be notified once it is done. A couple of announcements before beginning. Very shortly, the next big event will be Water Micro 25 in Amersfoort, Netherlands, organized by the team in the Netherlands. It's an IWA conference, a thematic conference every second year around water microbiology, health-related water microbiology, and of course we will also talk maybe a bit about sensors during that conference. So very, very interesting conference to attend. I'm sure some of you will be there. And then very recently this year, we have published a new critical review paper on automated on-site monitoring tools. It took a while to finish that paper, but it's done now and it's online, so you can have a look at it. It aims to make really the state of the art, and but critically also about the different tools that exist and what has been really used concretely for operational monitoring, how does it work, and what is really meaningful. So I think it's a good milestone for this new thematic. So coming back to the technical day, to the webinar, sorry. First session will be chaired by Nikki here on my side, Nikki van Bel from KWR. We have 4 presentations on recent case studies on microbial sensors, and we will start then with Alexander Kepsner. Just let me remind you that we have 15 minutes talk plus 5 minutes questions for this session. So I ask you please to keep the time at 15 minutes presentation. And Nikki, I hand over to you for the first presentation. I will share the slides.
You put your mouth. Yes, okay. Thank you for introducing me. So I'll be the chair of the first session, where we have 4 presentations of scientific researchers, which will share their results on work with sensoring in drinking water. So we have 4 presentations. After the break, we'll have session 2, in which we have 5 flash presentations of sensor technology suppliers, but it will be after the break. So we'll start first with Alexander Kirchner. Where is it? He acquired his master's degree in botany and microbiology at the University of Vienna in Austria. And during his PhD thesis, he worked on developing and applying methods for estimating bacterial abundance and growth in a natural environment. After having acquired his PhD degree in 1999, the focus of his research at the Medical University of Vienna and the Veteran Medical University of Vienna shifted towards the environmental occurrence, behavior, and fate of health-relevant bacteria in groundwater, drinking water, surface water, and wastewater. After becoming an associate professor at the Medical University in Vienna, leading the Water Microbiality Research Group at the Institute, he also became vice chair of the Inter-University Cooperation Center of Water and Health in Austria. Now, since 2018, he is also affiliated as associate professor at the Karl Landstein University of Health Sciences in Krems in Austria. With that introduction, it's now turn for Alexander to tell us all about the near real-time flow cytometry for operational microbial monitoring of alpine karst drinking water resources. So, Alexander, the floor is yours.
Thank you, Nikki, for your kind introduction and abundant introduction. I also want to thank Jean-Baptiste for giving us the opportunity to present this study. Now I have to switch slide. Yeah, okay. First of all, this is a short look back to the MS4W event in December 23. I guess you were all there, except me. Here you can see my main co-authors, Andreas Vanleitner and Lena Campostrini, who gave the introduction to this topic already. Andreas apologizes today because he cannot participate because he has a lecture at the Karl Landstein University. So his best greetings. Yeah. I want to start my talk with the history on microbial near real-time monitoring of karst springs. We understand near real-time monitoring is measurements below 1 hour, intervals below 1 hour. This can certainly be discussed. I read in your paper, Jean-Baptiste, that you define it differently, but we can discuss it later. So we began already in the 2000 years with real-time monitoring of turbidity and SAK 245 nanometers, and this was published a few years later. In 2010, we started with enzymatic measurements with the Coligat instrument, and this was published in 2014. And in the 2020s, we started with near real-time flow cytometry, and actually this is the data that I'm presenting today. Alpine karst springs are highly dynamic ecosystems, and they consist actually, this rock of these karstic systems, they consist of several zones. We have a saturated zone and an intermediate zone and an unsaturated water zone. And during basic flow conditions, the spring water quality is usually quite good and has a high biologic stability. but during precipitation events, sorry, I have problems to move. Yeah, during precipitation events, there is a substantial surface associated influence when the soil cover actually is moving down into these fissures of the carbonate rock and comes then into the spring and to the outlet of the spring. And the surface associated influence also transports microbes from the soil, also from potential surface water if they are there, and also microbes from fecal pollution. And this is actually the problem that we have. Yep. In the past decades, we have established a 3-step microbial management strategy for these alpine springs, together with the water company. And as a basis, step 1 consists of monitoring of fecal contamination. So traditionally, this is done via standard fecal indicators, SFIP. But here also online monitoring tools can be used. Online monitoring of microbial targets or also of surrogate targets like SAK or turbidity. And actually, these parameters can be used then for spring abstraction management. That means if the spring is used or if the water is discarded, if the values are too high. The second step is the characterization of fecal contamination in the catchment. So it's a pollution source profiling. Which pollution sources are there? Are they from animals or are they from humans? And this can also be supported by measuring genetic microbial source tracking markers. And actually, this leads to management measures such as catchment management and pollution control in the whole catchment. And the third step of this microbial management strategy is the health risk assessment of the fecal contamination, actually based on quantitative microbial risk assessment, for example, also to then calculate the treatment necessity and disinfection requirements for treating the water. Okay. Next step. Yep. Sorry. Yeah. On top of that, there are some publications on using flow cytometry for near real-time monitoring karst springs. Actually, all of them come from the James group, or at least Frederick was involved in these publications from Michi Besma and colleagues. They investigated 2 springs in a karstic region in northeast of Switzerland, and they evaluated different monitoring strategies to detect precipitation-induced microbial contamination events by using flow cytometry, and they linked total cell count to standard fecal indicator concentration and precipitation data. In this publication, in another publication, 3 springs in Jura and Western Switzerland were investigated, and they used near real-time flow cytometry, so the total cell count parameter in combination with other real-time physicochemical parameters, turbidity, SAK, conductivity, pH, to study microbial dynamics, and they linked also this near real-time and real-time data to precipitation and discharge data. And in these studies, they used the Akuri C6 flow cytometer with an on-site automatic staining module. The aims of our study was to test the applicability of a fully automated near real-time flow cytometer. You all know it. The BactoSense from Binovate for operational monitoring of an alpine karst spring. We measured total cell count, intact cell count. Also, we had data on the percentage of high nucleic acid cells, and also we used flow cytometry fingerprinting data. The second was to evaluate the indicator function of these flow cytometry derived parameters in comparison to the traditionally used physicochemical parameters, turbidity and SAC, towards surface-related contamination and fecal pollution. And the third objective was to establish early warning tools based on machine learning, which included also this fingerprinting data to predict fecal contamination in the spring. We applied a nested sampling design for this data series. So we had a long-term series on this data from 2012 to 2022 to identify in this catchment the periods of the highest fecal contamination potential, so the periods of the highest relevance for our problematic. And we added high frequency and event sampling in 2020 and 2021, actually during summer events. The parameters that we investigated were real-time abiotic parameters, as mentioned before, hydrology data, discharge and precipitation, then near real-time flow cytometry, total cell count measured in 1 hour, 4 hour, half an hour intervals, intake cell count, and so on, fingerprint data, a so-called dissimilarity score. And we also had high-frequency grab samples in the interval of 4 to 6 hours, adjusted to the sampling intervals of the flow cytometer, cultivation-based for total coliforms, indicating surface input, and Escherichia coli for fecal pollution indication, then DNA-based microbial resource tracking markers, all bug also for a certain surface input as a surface input indicator, and bug R as a ruminant-associated fecal pollution indicator for cows and wildlife. And also we measured parallel samples in the lab by fluorescence microscopy and flow cytometry as a reference, and to see if the BactoSense gives reliable results. And finally, yeah, we established these early warning tools, machine learning tools. Okay. Yeah, this is how the spring looks like. This is actually the building where the spring is collected. And here you can see a bridge where the instruments were installed. Here you can see these two BactoSense, one measuring the total cell count, the other is measuring the intake cell count. And then there is also the water pumped out from this spring building up to the bridge with a hydrocyclone to prevent a bubble to prevent bubbles. And also this water is then transported to an autosampler where the samples were taken for cultivation-based and DNA-based determination of fecal contamination. Here you can see also a field lab where we did the DNA concentration and the SFIP determination nearby. Yeah, so this is the results from the long-term monitoring. Actually, what you can see here is that the highest discharge usually occurred during the snow melt, but also high discharge events occurred during the summer period. And the fecal pollution events that you can see here in panel D they only occurred during summer. That means between June and November, and not during December to July. Sorry, to December to May. Also, the microbial resource tracking markers occurred only during this period. So the ruminant marker concentrations only had peaks during the summer period. Okay. Yeah, this led us to sorry. Yeah, during the high frequency and event sampling in 2020 and 2021, we found 6 events, actually 3 events in year 2020 and 3 events in 2021. So 6 events were identified, and all of these events could be identified by the cell count methods, total cell count, intake cell count, and HNA%. While, for example, the SAK and turbidity did not reliably detect the event 6 in the year 2021. The fecal pollution data but clearly showed that this was really an event where we had a big increase in E. coli and also total coliform counts, and also in BAC-R concentrations. And also the dissimilarity score based on the flow cytometry fingerprints detected these events here in dark red. You can see that the event alert actually was caused, but event number 1 with this dissimilarity score was not detected. Next slide. Yeah. Here we were looking for the indicator function of the flow cytometry derived parameters in comparison to the physicochemical parameters. And here for this comparison, only the data from 2021 were included because we had enough data in this year in comparison to the year before. You can see that the data set was that we had much more data in 2021 than in 2020. And that's why only this data were included. And surprisingly, we had a similar high correlation of the fecal pollution and surface contamination with the chemophysical parameters and the flow cytometry parameters. So these parameter types performed actually equally. Yeah. So what was also surprising was that the relationship of the flow cytometry data was higher to the fecal indicators than to the indicators for soil and feces. And for this, actually, so far we do not have any explanation, because the bacteria that coming in that you're measuring with the flow cytometer are not specifically from fecal pollution. They're coming from soil, fecal pollution, surface waters, and so on. So they cannot discriminate between these different sources. And it's funny to see that the fecal indicators were more significantly and higher correlated to the cell counts than the general indicators of soil and feces and surface contamination. Yeah, concerning the applicability and validation of the flow cytometer, there were some technical challenges on site. We had a high humidity and a low temperature in these spring buildings. Actually, we needed about 30 minutes of warm-up time of the instrument, but later we had no problems during the whole investigation period for several months with the BactoSense. It worked throughout the whole investigation period without any problems. The only thing that sometimes occurred was some power failures, but now with the new software, this is also solved because the BactoSense is now able to restart automatically and doesn't need to be restarted by a person. Yeah, and the continuous water supply without bubbles can be solved with a hydrocyclone, so then also the instrument can be run through a long period without any problems. For the comparison of the new real-time flow cytometry data, so the on-site data with the fluorescence microscopy data and also with flow cytometry data in the laboratory, we created here a band oatmeal plot showing the variability between the different methods. What you can see here is that the variability is actually evenly distributed. Sometimes the fluorescence microscopy overestimates the cell number in comparison to the flow cytometer, and sometimes it underestimates. And we found out that this is a non-random distribution of the differences for each event, because it shows that for specific events there is an underestimation of flow cytometry, and for specific other events there is an overestimation. So this is maybe owing to the background metrics of the specific event, leading to these differences between the methods. But all in all, they gave quite reliable, comparable results. Yeah, concerning the early warning tools to predict the contamination events, we applied 2 machine learning models, and the random forest classifier is a model that performed the best. I don't want to go into detail, but all these relationships and also these prediction models for E. coli, bug-R for the specific fecal pollution indication, and also for coliform and all bug, so the surface water prediction, all performed best when flow cytometry parameters were included, and not only the turbidity or SAK. I'm already coming to the conclusions and summing up of the talk. Yeah, concerning the practical performance of the automated flow cytometer, the validation results showed that the BactoSense delivers reliable total cell counts and also intact cell counts. No problems were observed with the high humidity and low temperature in the spring. The specific technical equipment is necessary to guarantee continuous bubble-free water supply to the instruments. This is very important. Concerning the detection of the contamination events, the events showed different characteristics. Specifically, event 6 was completely different than the other events, and in real-time flow cytometry could reliably detect all the 6 contamination events, not all parameters of them, but at least one parameter. And event 6 could not be detected by the chemophysical parameters. Yeah, concerning the indicator function of flow cytometry derived parameters by the correlation analysis, a similar performance as the chemophysical parameters, turbidity and SAC actually occurred. And the machine learning models showed that all models containing at least one flow cytometer parameter performed the best. And what are the implications of these findings for the water management? Actually, we think that the flow cytometer provides valuable additional information on pollution events in karstic springs. It can be a useful tool for operational monitoring of karst water resources, for example, for triggering an autosampler, or also as decision tool for spring abstraction management. However, this is a study only done on one specific spring and cannot be extrapolated to other springs. We have to test it in other springs, and we are currently testing it in other springs as well. So the usefulness, the general usefulness has to be tested more generally in different catchments, also in different springs. And with this, I want to say thank you for your attention. I also want to give special thanks to my co-authors, specifically here to Katalin Demeter, who I didn't mention before. She's also a co-author of this study, a main co-author of the study, and thank you again for your attention.
Thank you, Alexander, for an interesting presentation. Are there any questions? You can put them in the chat. Sorry? Oh.
I hope I kept the time. I'm not sure about that.
2 minutes too long, but
17 minutes instead of 15. Okay, that's quite good.
Yes. We have time for a few short questions. I do have one while all the others can think of a question. You mentioned that the fecal contamination, that the fingerprint of the flow cytometry is enough to detect fecal contamination. Is that right? Or do you need the other parameters?
No. Actually, this is only correlation analysis. Yeah? We can only link the contamination event, the fecal contamination event, with an increase in cell counts or a change in the fingerprint. Yeah, you will never be able with this method to directly detect fecal contamination. This is really a misunderstanding. Yeah, you can only see a correlation, and in the sorry, in the machine learning models and prediction models, this parameter often was a good parameter to predict the fecal contamination, but you can never put it one-to-one, because total cell counts is a general cell count parameter, and fecal contamination is very specific. Yeah, we would need actually a specific cell stain for E. coli that can be sensitively detected with the flow cytometer, but so far this is not available. Yeah, maybe we should look for targets that are higher abundant. Yeah.
I have one question in the chat that we can do. The question is, how is an event defined? How do you know if it's an event?
Sorry, I didn't understand.
How is an event defined? So how did you define what are the events with the data that you had? What are the characteristics of an event? How did you set the boundaries?
I cannot understand your voice. I'm sorry, it's not good understandable for me. I have it.
You cannot hear me anymore?
Yes, now it's better.
Okay, I don't know what was wrong. I'll try again. So the question was, how is an event defined? You had 6 events. How are they defined? What are the? How do we know that?
Yes, the definition here for this study was that the precipitation is higher than 30 millimeters per 24 hours, followed by an increase in E. coli or fecal indicator bacteria. So this was our definition of the event. Can be defined in another catchment or in another study differently. For us, precipitation, increase in discharge, and E. coli increase had to come together. Yeah, because you also had, let's say, increase in discharge after snow melt, but there was no increase in E. coli because there were no fecal pellets available for being moved into the karstic spring. Yeah.
Okay, thank you. I think we have to go to the next one due to time constraints.
Okay, thank you.
So you hear me? So we'll continue. I will be presenting Nikki for the next talk. So Nikki is a researcher at KWR Research Institute in the Netherlands in the field of microbiological water quality and health. She is active in several projects concerning new rapid microbiological methods or sensors, and that were tested in a laboratory, drinking water field tests, and non-drinking water field tests. In this, she has compared sensors to operational procedures of the drinking water production plants, conventional microbiological measurement programs, and she studied what the added value of sensors can be in the water industry, both for the Dutch drinking water companies and other water-related areas. The major goal of these studies is to improve and monitor the microbiological water quality of drinking water recreational water and other water types. And today she's going to present the alternatives for HPC22 in the drinking water distribution system after repairs. The floor is yours.
Thank you. Just wait for the slides.
Hand over.
I'm still seeing the other slides.
Oh, sorry. Now should.
Yes, there we are. So I will present our results that we published roughly half a year or a year ago. It's about an alternative parameter for heterotrophic plate count in the drinking water distribution system, especially after repairs. Yeah, so well, as everybody knows, if there's a maintenance or pipe breaches in the distribution system, you run the risk of microbial contamination of the drinking water. So in the Dutch legislation, the procedure is included what to do after repairs or after pipe breaches or calamities. So first, when the pipe is intact again, you first flush with 3 volumes with 1.5 meters per second. Then after 1 to 24 hours, we take samples. And we analyze them for E. coli and enterococci to test for fecal contamination. And we also have heterotrophic plate count at 22 degrees to test for microbial contamination in general. And with that last parameter, we specifically detect cultural bacteria that grow on high nutrients level. And those are the ones that you expect to be in the water if there's a contamination from outside. And if these analyses are okay, the drinking water company can resume the water distribution. Let's see. Go. Yeah. It's not very surprising, but E. coli, enterococci, heterotrophic plate counts are culture methods. So you have to wait 1 day, 2 days, or 3 days before you have a result. And the drinking water utilities in the Netherlands were not very happy with that, such a long delay. So we have been busy in the last 10, 15 years to come up with alternatives to shorten this period of time that they have to wait. So something is wrong. So the enterococci method, we developed an RT-PCR method that was implemented last year, 1 year ago. We also, oh, I'm missing one. Where is it? The E. coli sorry, there's something with my slides. The RT-PCR for E. coli was implemented in 2019, the enterococci RT-PCR in 2024. And now the last parameter is heterotrophic plate count. And the question is there an alternative method available that's faster and just as sensitive or sensitive enough to replace heterotrophic plate count? And we're really looking only at the situation after work activities, after repairs in the distribution system. So really for that specific situation. We compared heterotrophic plate count to 5 different methods. So we had, of course, the culture method, heterotrophic plate count. We compared it to ATP, the lab method at KWR, and the mobile luminultra kit that you can perform in the field. We compared it to laboratory-based flow cytometry for the intact, non-intact, total LNA and HNA fractions. We compared it to the sensor, 2 sensors that measure enzymatic activity, the bug control and the coliminder. And we compared it to a molecular method of 16S RNA gene with the qPCR. And this is a short overview of what we did. So we started with artificial contaminations. We started with drinking water, and we added several contaminants to the drinking water. So that was either surface water, rainwater, sewage, sediment, groundwater, or clay soil. We made different dilutions. We measured that with all the different methods that I just mentioned. Afterwards, we went into the field. So we asked for drinking water utilities whether when they would have repairs of the distribution system, so that we could come and take samples before and after. And we also asked them to inform us if there was a calamity that we could come and sample, because those are the most high risk situations. So the artificial contaminations, we did 12 dilution series. We had 2 non-chlorinated drinking waters in which we diluted 6 contaminants. So wastewater, surface water, rainwater, sediment, groundwater, and soil. And we chose them because those are the ones that could contaminate the drinking water in the distribution system if indeed the ground, if the pipes are open in the distribution system. And we measured all these dilutions with all the methods in parallel. But first, it's just an example of these two graphs. We have way more graphs, but then it's better to read the paper instead of showing them all here. So we had first looked at how sensitive the methods are. So on the top, you can see the heterotrophic plate count. We made 9 different dilutions, and here we can still count heterotrophic plate count in the sample at dilution number 7, but at number 8 we don't see them anymore. If we compare that to the flow cytometry, then what we lose the signal at around 4th dilution, 4th or the 5th dilution. In the beginning here you see a very nice decrease in the number of cells, but afterwards it becomes flat, which shows that in this case the heterotrophic plate count is more sensitive in detecting a contamination in the drinking water compared to flow cytometry. And flow cytometry was not the only one. For example, we also had it with we saw the same situation with the 16S RNA gene, which also showed the same pattern. And this is caused by the high background level in the drinking water. So in the Netherlands, we have a high number of cells in several different distribution systems. So if the number of cells is already quite high in the drinking water, you don't have that much space left over to really detect contamination. So if we zoom in a little bit more to the results, so here I compare heterotrophic plate count on the x-axis to ATP on the y-axis. And you can see a very nice correlation for all of the different ATP parameters. So on the left, it's the total ATP from the laboratory method. In the middle, it's the mobile ATP kit. Sorry, on the right side is the mobile ATP kit. What we noticed is that ATP is the most sensitive alternative method. So heterotrophic plate count is still a little bit more sensitive than ATP. So, but ATP is the second best and still very good and good enough for our purpose. So it could detect the lowest percentage of contaminant after the heterotrophic plate count. We have a very nice correlation with a good R-square of about 0.7. And all contaminants that we use, so all 6 contaminants, we could really detect with the ATP method. If we compare the heterotrophic plate count to flow cytometry, so on the top left, this is intact cells, total number of cells, LNA and HNA fraction. You can already see that the correlation is way less compared with the ATP, but that's not very surprising. It has been shown before in literature that the correlation between heterotrophic plate count and flow cytometry is not always that good. Can be really bad, can be good, but it varies. But more importantly, it's also that the flow cytometry is not sensitive enough, and you can, for example, see that here. So here in the top, the higher concentrations, we have quite a good correlation. If we go to the lower concentrations, this flattens out. So we do have lower numbers of heterotrophic plate count, but we don't see that decrease anymore with the flow cytometry. And on top of that, the flow cytometry could not detect contamination with rainwater or clay soil. We compared it also to the enzymatic activity. On the left side, the bug control, on the right side, the choline minder. For the bug control, we did not have a very good correlation between the two methods, and also the system was not sensitive enough for our purpose. The choline minder had a very good correlation of 0.7, so comparable to the ATP method, and also was sensitive enough for our purpose. However, both methods did not detect rainwater and clay soil, so it's really the difficult matrices for all the methods basically, and the bug control also could not detect clay soil. So the last comparison from all the artificial contaminations is the 16S RNA gene. Also here, it's not sensitive enough. You can see it again here on the left bottom side. We do have lower numbers of heterotrophic plate count, but we don't see that decrease with the 16S RNA gene. The R-square is quite okay, but that's mainly caused by the higher numbers of heterotrophic plate count. And also this method did not detect a contamination of rainwater. So to conclude, what came out from the solution series is that the heterotrophic plate count is the most sensitive method. It detected the lowest concentration of a contaminant, followed closely by ATP. And then the coliminder also very closely behind that one. The ATP lab method and the mobile method from Luminultra and the coliminder seem to perform best in our laboratory experiments, whereas the flow cytometry and the 16S qPCR suffered from high background levels in drinking water, so that because of that they were not sensitive enough. And concerning the enzymatic activity, the two sensors that we tested, the bug control seemed less effective than the coliminder. So then we went. Okay, I go to the next slide, I hope. Yeah, so then we went into the field. We went to 6 different distribution systems where we sampled after repairs or after calamity. So we had one calamity, the number 3, a leaking fire hydrant, which had to be replaced. We had 3, we also had here one calamity, and all the others were planned replacement of certain parts in the distribution system. So that was easier for the planning of our laboratory. But still the calamities we would have liked to have more, but that's a good thing that they don't happen that often, but for the research it's always a little bit challenging. So we went into the field and we took 9 samples in total around a calamity or a repair. So we took 1 sample before the maintenance work was performed, or we, in the case of calamity, we took the sample upstream. And then we took samples after flushing, after 1, 2, 3, or 5 hours after flushing. And then on day 2 and day 3, we also took samples. The last sample we took was after 48 hours after flushing. And these are all the results we got. And you can look at all the graphs, but I can also shortly summarize, which means that you can also look at the correlations, but you can see the dots are very nicely scattered around the graph. The problem with our field studies is that the heterotrophic plate count hardly ever exceeded the 100 CFU per milliliter, which is the legal value in the Netherlands. So we had hardly any aberration of the drinking water qualities with heterotrophic plate count, but also not with alternative methods. So we were measuring at relatively low heterotrophic plate count levels, whereas we were interested in both low but also higher levels. So based on these results, we cannot say in the field which of the method is the best one and which should we implement in practice. So what we do see sometimes that heterotrophic plate count is a little bit more sensitive than the alternative method, but still it's based on 6 locations. So I wouldn't put my hand in the fire to really draw this as a very strong conclusion. We couldn't find any contamination of the field samples. So like I just said, we don't have a clear picture from the field samples on how the alternative methods performed. To do that, we need further field studies, and that's not only 6 studies, but we would like to set up a national database, not a database, but national study to include way more of these repairs, all these calamities, and then compare the methods to each other. So that brings me to the overall conclusion. So for the artificial contaminations, heterotrophic plate count was the most sensitive method, detected the lowest concentration of a contaminant. Of the alternative methods, ATP methods and the coliminder also detected a very low contaminant concentration, followed by flow cytometry, 16S, and the bug control. Heterotrophic plate count and ATP detected all contaminant sources, except for rain in some cases, whereas flow cytometry, 16S, bug control, and coliminder did not detect rain and or clay soil. So we saw the strongest significant correlation between heterotrophic plate count with ATP, the lab method, the mobile method, so the field method, and the coliminder. And for the field samples, it's a very short conclusion that we cannot draw a very strong conclusion. We need more field samples and preferably also with higher heterotrophic plate count numbers. So that's what I just mentioned. And it brings us to, well, now, what about implementation? So because of the lack of variation in heterotrophic plate count in the field samples, we need to do more of those comparisons. So we would like to do an extensive comparison between heterotrophic plate count and alternative methods. Based on, I haven't showed you that today, but based on the performance characteristics, cost, analysis time, ease of use, and robustness, we would like to suggest to compare heterotrophic plate count to ATP methods and Antarctic holy minder. So by doing that, you would take samples at all the repairs in the Netherlands and in Belgium at the Watergroep, and measure them with heterotrophic plate count, ATP, and holy minder, and see which method is the best to implement, hopefully in the future in the legislation. So, and if we have that database, we can also set signal values. We know it's 1,000 CFU per milliliter for heterotrophic plate count, but we also need to set those guidelines for the other methods. So basically, we've got work to do. And that brings me to the acknowledgement. So the research was part of the joint research program for the Dutch drinking water utilities and the Flemish drinking water utility, the Watergroep. And I didn't do this, of course, alone. I did it together with Frits and Marcella a lot, and my other colleagues. And you can see a very nice picture of 2 weeks ago where we had a nice team day. So that's it.
Thank you very much, Nikki, for this interesting presentation. We are on time, so we have fully time for questions. I don't think I see any here in the chat for now, but just drop in your questions if you have some. I would like to ask you then the first question. For the lab work, you said that you had some better correlations with some technologies than others. In terms of risk, how should we address this also in terms of risk? Because we are going more and more to risk-based approaches with these water regulations. If you detect something up to a certain limit, how would you deal with the risk?
I think until now that would be the same as how they used heterotrophic plate count. So they have to measure 3 parameters: E. coli and Enterococci is fecal contamination. So if they find those, they will never release a drinking water pipe into. They will parameters. However, with heterotrophic plate count, it's already different. Where they it should be below the 1,000 CFU per milliliter, but it's not a very strict guideline in that if it's above, they cannot distribute the drinking water. So they have to flush again. They have to take extra samples, but they can start distributing the drinking water as long as the E. coli and Enterococci fulfill the parameters. So the heterotrophic plate count is less strict parameter than the other ones, but they will keep on trying to get the drinking water below those 1,000 CFU by extra flushing and taking extra samples until it is okay.
Okay, thanks a lot. Response. So I have one question here in the chat. So there is one participant asking, for what enzyme did you use for the enzymatic activity measurements? Was it glucuronidase, galactosidase, or any other?
Yeah, I didn't mention that. We used alkaline phosphatase, so that's the parameter for general, the total bacteria that are present in the drinking water.
Mm-hmm. Which is also closest to ATP, I guess. Another question here in the chat: how is the value of 1,000 CFU per ml defined?
It is from the Dutch regulation. We tried to look back, but in the history of how it got in there, but as far as I know, we couldn't really find a result. It is true that in European legislation or worldwide, it often is around that value, but I have to admit I don't know the exact answer to that one.
I think there's still time for one question. Have you tried or considered simple tryptophan fluorescence as a potential surrogate?
No, we have not. Well, we have no experience with tryptophan. And what I've always understood from tryptophan fluorescence is that you need to know your drinking water very well before you start using it, and you can indicate when it's a deviation and when it's still normal. And in case of repairs, you cannot set such a baseline. So, and that's why we did not include it, one of the reasons.
All right. Thanks a lot for everyone. If you still have questions, keep them, and you may also send them to Nikki. Also can refer to the paper that was published recently, as you can see here. So I hand it over back to you as chair.
Yeah. So we continue the webinar with the third presentation, which is from Fien Wagenaar. She's a passionate last year PhD student at the CIMET of the Ghent University in Belgium, specializing in the dynamic field of drinking water microbiology. In 2020, she graduated in bioengineering from Ghent University, where she conducted her thesis with a focus on drinking water biostability, earning the Water Technology Award for the best master thesis. She continued in Ghent for its PhD researcher, contributing to the Biostable project, focusing on drinking water biostability. And today she will present to us the research finding, she presented the research finding at various conferences, including 2 Young Water Professional conferences and the Biological Society for Microbiology conference in 2023. And furthermore, she's had the privilege of mentoring and guiding 8 different students, and she's actively engaged in educational activities, particularly in the domain of microbial growth kinetics. So today, Fien will present on the flow cytometric fingerprinting as a tool to evaluate and predict water quality changes in practice. Fien, the floor is yours.
Yes, thank you, Nikki, for the introduction, and also thank you for the opportunity to present part of my work. So today I will talk about a case study that I've done together with a water provider, PITPA. And PITPA is a water provider located in Antwerp in Belgium. And I will focus on flow cytometric fingerprinting, as Nikki already said. Yeah, more specifically fingerprinting and not the cell counts, because Nikki already told in her presentation that cell counts are maybe not sufficient to detect low concentrations of contaminants. And the case study was really to evaluate flow cytometric fingerprinting in practice. So to see in which cases is flow cytometry now of added value, and in which cases is it not of added value. Okay. So very short, some background information of the water company PITPA. So they are doing routine monitoring at regularly time points and at regularly bases and places. And they are measuring the legislative parameters, total coliforms, heterotrophic plate counts, and so on. But next to that, they are also having a flow cytometer in their lab. It's an Acudi flow cytometer. And they are measuring offline some samples with flow cytometry. The data of that analysis is then put at a platform that is performed by Kitos. It's a spin-off from the UGent, and they sell the platform to do data analysis actually, and to perform the data analysis and to show PITPA then the cell counts, the diversity matrices, and so on per region. So it's actually summarizing all the results of all the flow cytometric data. But which samples are they analyzing for flow cytometry? So they were taken last few years samples of certain water towers that have quality issues, also of their production centers. And also when there is a contamination event, they also starting to take samples of flow cytometry to learn more about the contamination in combination with flow cytometry. So the platform is just actually a platform with numbers, and I went a bit deeper. So I used the data to see where is flow cytometry of added value and to do some more data analysis actually. So this was a bit the goal of the case study. Now very short flow cytometry for the ones that do not know. So flow cytometry is an optical technique. You have a water sample, you add a dye to the water sample to stain your bacteria in the water sample. Then your water sample is introduced into a system. The cells are passing one by one through a laser. That laser is causing an excitation and an emission of light at different wavelengths, also different scatters. So a lot of information that is measured then by detectors, for example, fluorescent detectors that are measuring at specific wavelengths, but also forward scatter detectors, cyber scatter detectors, and so on. So actually, then you have fingerprints, because every water sample is unique. Every water sample will cause scattering in different channels and so on. So this made that you have a unique fingerprint for each water sample. And based on that fingerprint, so I present it now like this. For example, a parameter from a detector, red fluorescence, for example, a function of green fluorescence, for example, another parameter that is measured by the machine. And on all those parameters, you can do a kind of a binning strategy, as is shown in the second part of the figure. You can do fixed binning strategies or adaptive binning strategies, a lot of possibilities actually. And then you have a huge data set. So for each water sample, you have the amount of counts per bin actually. And that is unique for every sample. For example, sample 1 will have a lot of counts in the first bin, but less in the second bin and so on. And this made that you can do a lot of statistical analysis and additional analysis to see if samples are similar or not. And that's the analysis that Kitos that is presented on the platform. But I went a bit more robust actually. I've done a similar analysis, but on the data set, on the large data set, I've done a kind of a cluster analysis. And that cluster analysis is simply, it goes over your samples, and then it says like, okay, this sample is more looking like that sample, I will put it in cluster 1, group 1, for example. That sample is more similar than that sample, I will put it in group 2. And the analysis is making groups, clusters actually, sometimes 2 groups, sometimes a lot of groups. And the goal was, can we now distinguish a group, a cluster 1 that is, for example, only containing bad samples, and cluster 2 that is only containing good samples. So that's the analysis. So it's a very robust technique that is dividing your samples in multiple groups. So I hope everyone is following a bit, but we tried to do this kind of analysis on the water towers, on the contamination events, and so on, to see where it's working and where not, because it gives the opportunity actually to train models in the future based on contamination events. That's a bit the goal, the further goal of the case study. So I will go over one case study that was successful, and it's a case study about a rainwater intrusion event. It happened in January 2024. So about the case study itself, what happens? So there are some streets in Antwerp, as you see on the map on the right, and there was a misconnection with the internal rainwater circuit. So the house that is located in the red streets with the red circle had a misconnection, and there were complaints in the surrounding streets about the quality of the water. Okay, a bit about the intervention plan. So complaints about a water sample, they call the water provider comes to take samples. They see that there is something wrong. So they start an intervention plan. They take extra samples, start controlling installations, because it was actually, there were a lot of new houses in that neighborhood. And they start flushing and so on. In between, they also took a lot of sample for flow cytometry. Then a few days later, 4 days later actually, the misconnection was identified, and they also took a sample for flow cytometry of the raw rainwater, so of the really bad sample, which is really good for the analysis, because you can train, you can train the model because you have a really bad sample, which is good. And then the intervention stopped. So they had also analyzed a raw rainwater sample. Of course, also the legislative parameters were measured. And as you can see, it was really a bad sample. There was Iromonas species present, Enterococci, E. coli, some E. coli were present, total coliforms were present, so it was a bad sample. And it was really good that we had this bad sample, because now we can see, okay, this sample will belong to a cluster. Which other samples are belonging to that cluster? And is it logic that they are in that cluster or not? So let's go to the results. So here for the red street, I'll start with the red street, of course. You can see the cluster type is just 1 or 2, because the analysis was able to define 2 clusters instead of more clusters, which is already easy to understand, in function of time. First of all, you can see the really bad sample. It's the cross. Maybe have a look if I have a pointer. Okay, so the cross in green here is the raw rainwater sample. That raw rainwater sample has 200 coliforms in it. And you can see the other samples from that street. So there were a lot of samples in that street taken in function of time. You can see that before, so here at that moment, the problem was solved here. And you can see that actually before, there were a few bad samples regarding our analysis. So there were samples that have coliforms. The triangle, for example, had 1 coliform, but also another sample that people thought that there was rainwater in it, and also our analysis saw that there was rainwater in it that also belonged to cluster type 2. Other samples belonged to cluster type 1, so actually the samples of good quality regarding our analysis. But the analysis was not always good. For example, a sample, that cross here, a sample with 4 coliforms was also classified as good. But this is a bad point of the analysis. But what is good, that it's after flushing and after identification of the misconnection, samples were always having cluster type 1, except an outliner here. But it's a good start, actually. Let's go to other streets to compare. For example, the green street that is located here. That was already better, actually. So you can also again see cluster type 2 is again the bad cluster, because we did the analysis on all of the samples. Cluster type 1 is the good samples. And you can see that now the analysis was able to take out all samples with coliforms, as you can see here. Then after the mixed connection identified, samples were good. Here there was still a bad sample, but that sample also contained coliforms, so maybe there was some left over in a piece of a house or something. So for this street, the analysis was quite good to distinguish good and bad samples. Then for other streets as well, another street, for example, that street here, was not affected regarding coliforms, but also for the analysis, samples were also classified as cluster type 1, so that street was not affected, actually. This is again the raw rainwater sample. And then for another street, you can see that, so the blue street, the orange street, again, the cluster type 1 and 2, the raw rainwater sample, and then there is one sample here that has no coliforms, but regarding the analysis, regarding the microbial community, there could be rainwater in that sample. So actually, the analysis was quite good in distinguishing good and bad samples, but it's a really specific case. And for some streets, the analysis did better than others. So let's go back to the future perspectives and to what we have learned now. So we see that we have a defined region here. We have a very short time frame, only 5 days. But we saw that there were some microbial dynamics detected with our analysis, detected with flow cytometric fingerprinting, related to water quality shifts. We also saw that the analysis, the cluster analysis, was able to separate samples with rainwater for some streets. What is coming? And we saw that sometimes we saw that a sample was bad, even though there were no coliforms detected. And the good thing about the case study was that we have the raw rainwater sample, we have a bad sample, and we have clean samples, so we can compare. But it's a small data set to train the model, and it's a very specific case. But it's a food for thought for next case studies and next things. Because for the other case studies, we saw that, for example, the water towers, that's a lot of different other factors. For example, they clean the water towers, they chlorinate the water towers. So the microbial community is influenced by these events as well. The analysis can detect it, but there is too much happening, so it's not robust anymore. Also, we saw that while analyzing all the data, that it's needed to have a fixed sampling pounds, but also good periods, because now we only have the contamination events. We don't have any samples from before to have a robust baseline. You need relevant metadata. And now the next step of PITSPA now, together with Kitos, is to use machine learning to develop an algorithm to track microbial changes as a quality indicator. So they are now going a step further. They are testing in the lab also contaminants and doing that kind of analysis. And they will now focus more, because that we saw that with the water towers it was not working. They will now focus more on regions that have regularly quality issues. And they are taking now already samples from those regions before. So yeah, thank you, of course, Pitpa and Katrien. She is the head of the bacteriology lab, and she helped quite a lot. She was really interested in going further and going further by using online monitoring in the drinking water network. And also thanks to the promoters, to my promoters, Bart Hussem and Nico Boon. And thank you for listening. And if you have any questions, I'm happy to answer them.
Thank you, Fien, for more information about the flow cytometry fingerprinting. Every time I learn a little bit more about it, so that's good. Please post any questions in the chat. I will start with one, so you can all think about other questions. I was wondering, you already mentioned it a little bit at the flow cytometry and the other case studies. Does it also work very nicely if you don't have a bad reference sample, in this case the raw rainwater? Is it also applicable to those situations, or do you really need a bad sample in this case?
In this case, it made it easier to train the model to have the bad sample. But if you have a lot of metadata, it's also useful. But now we had the bad sample, which was most useful. We didn't have the good samples before. So then the bad sample is really useful. Yeah. But that was a bit a trade-off for PITSPA to know, and they want to go further. They want to learn from the flow cytometric samples that they have analyzed. And now we can go further with this.
Okay, thank you. Let's see, we have question in the chat. So for users starting to explore fingerprinting, do you have a recommendation about the channels you use to feed analysis pipeline? FL1, FL2, FSC, SSC. Do you have any tips for people starting with fingerprinting?
With the fingerprinting. Actually, I used 4 channels, FL1, FL3, and a forward scatter and a sideward scatter. But you can use more, and it's also depending on the dye and the stain that you are using. But if you are using, for example, the most common dye, cybogreen, you have a signal then in the FL1 detector, it's just the name of the detector, and the FL3 region, because you are measuring that wavelength. Then it's useful to take the results of those 2 detectors instead of another one that is measuring in another wavelength. So depending on the stain that you are using. But I mainly use 4 detectors.
Yeah. Okay, thank you. There's another one. Yeah, I was also thinking about that one. In a near real-time or in a real-time contamination scenario, how long does the post-hoc cluster analysis take?
The cluster analysis itself did not, no, it was not so long. I think it was, so we are working on a server also, so it's also a bit faster. but it's needed because the flow cytometric files are also very heavy to just analyze on your computer. It was 5 minutes or something, but it takes more to do the fingerprinting before. So preparing your data set takes a bit longer, and yeah, that's also needed, and you need a lot of samples already for that. Yeah, or you can do a fixed binning strategy, then it doesn't take long at all. But if you do adaptive binning strategies, so then the binning is done based on the events in a certain bin actually, then it will take longer. So it's depending on how much time you have and how much computer speeds.
But continuing on that, is it a suitable technique to really use in a contamination case or suspected situation that happens now? Or is it for now really looking back at what happened and can we explain things? Or do you think in the future, near future, that it would be more, I don't know, faster and that it's easier to really use the data at the moment?
I think when you have a model, for example, now it's specified for rainwater contamination, but if you have other contamination models as well, and you are regularly taking samples for flow cytometry, and then you see sometimes, you see at a certain moment, the contamination cluster popping up, the cluster 2 in this case, then I think it will give you fast information faster than the results of the plate counting. Because now, for example, they started taking samples of flow cytometry on the 16th, but then they knew that there was already bad things happening. But we also saw that on the 16th, samples were also bad regarding our analysis. So I think it just needs more time, more samples, more getting a bit more handy with the data and knowing what's in which cases it really offers added value, because in some cases it's not.
Okay, thank you very much for the questions also. Then we go to the last presentation of the first session, which is Jean-Baptiste, so the organizer of this nice webinar. So he obtained his PhD from the University of Liège in Belgium before carrying out a postdoc at Polytechnique Montreal in Quebec in Canada. Since 2020, he is working as a research and technology associate at the Luxembourg Institute of Science and Technology. His research has been focusing on source water protection and quality monitoring in a context of safe drinking water supply. And he has been investigating the spatiotemporal dynamics of waterborne pathogens, as well as harmful cyanobacteria and their toxins in water to help better assess and monitor associated public health risks. During his research stay in Quebec, he implemented automated online monitoring of microbial contaminants in drinking water supplies and recreational waters. He also actively contributed to the global effort of SARS-CoV-2 surveillance through wastewater in Quebec and both in Luxembourg. And today he will tell us all about the association between automated on-site monitoring of glucose activity and pathogenic protozoans in an urban drinking water supply in Montreal in Canada. So, go ahead.
Thank you very much for the introduction, Nikki. So let me see here. Okay. So I'll just here again cite some of the technologies that have been developed over the decade. There are multiple solutions now to monitor bacterial water quality. I say bacterial because it's essentially bacterial up to today. I mean, there is no tool yet to monitor viruses or protozoan pathogens like this. It's essentially bacteria that are being monitored with these methods that are fully automated. We've addressed some of them today already by our previous talks, enzymatic activity, ATP, flow cytometry, of course, and there are also others for culture and also direct imaging. They are measuring at high frequency in their real time also, and which is also important, is online. They can be accessed online, remotely controlled, and also the data can be visualized remotely. So the water regulations are now for a while now shifting more and more towards risk-based approaches. So these new tools can be a really added value to develop early warning systems and also improve risk assessment in the framework of more efficient water safety planning. And when you look at your drinking water supply system from source to tap, like here, you can do many things with these new technologies. I've summed up some of them here from our paper, review paper. If you look at source water treatment, distribution network up to the building, there are various applications that have been demonstrated, published, and there are also transversal actually applications like designing new monitoring strategies or also perform event-based sampling. We've heard today a talk from Alexander Kirchner to identify hazardous events, track fecal pollution sources, and also perform event-based sampling. And right now from Nikki, we heard about follow-up of water quality after maintenance in the distribution network. So there are already 2 applications covered today. And I'm going to talk also in the source about the source water, similar to the presentation of Alexander. In Karst here, it will be in surface water in an urban context. So we wanted to know in Montreal, this work was actually conducted in Montreal during my stay at Polytechnique Montréal. What is the vulnerability of a drinking water treatment plant intake to pathogenic protozoa, Cryptosporidium and Giardia. So we also looked at identify hazardous events, track fecal pollution sources, and perform event-based sampling for risk assessment. But we didn't do it with flow cytometry, but with another technology that I will present soon. And the idea was also to know, okay, what are the pathogens that are, so the protozoa that are detected during events of fecal pollution. So the urban river is in the Greater Montreal area. It's really a typical case for de facto wastewater reuse with succession of wastewater treatment plants, like here you can see the symbols, the white triangles that are then followed by drinking water treatment plant intakes, and in between you see a numerous CSO discharges. So there are more than 7 wastewater treatment plants and more than 150 CSO discharges over that 40 kilometers river. And the big dot here, the yellow dot, is the drinking water treatment plant that we have investigated first. And afterwards, I will also show you some results from a downstream drinking water treatment plant, the number 2. We conducted online GOOS activity, so glucuronidase activity measurements with the coliminder. Was measuring every 2 hours. You see here an older version of the coliminder because it's a couple of years already. And we were measuring every 2 hours, every hour during a specific event. And E. coli was measured every 3 to 4 days by the municipality routinely over 3 years. So the glucuronidase activity, I just summarized it here, the reaction. The enzyme catalyzes a reaction that converts the fluorogenic substrate into a fluorophore, and you know this from the E. coli alert assay to detect, to identify E. coli. It's the fluorescent wells that you can count. It's the same principle. There is no culture steps. So we have the result within 15 minutes because it's a kinetic reaction. So it's actually bypassing the cultural step, and so it's much more rapid. So the enzyme is actually it's kind of an index of a proxy for E. coli in water. We also collected municipal data on combined sewer overflows and bypasses from the wastewater treatment plants, as well as the local hydrometeorological data. So for pathogen measurements, we measured Giardia and Cryptosporidium. They are protozoan parasites using the USPA method 1623. And during those events that I will show you now. Here's the dynamic of the glucuronidase activity over this monitoring period from 2016 to 2019. And you can really see there is a seasonal trend with vulnerability periods in fall, winter, and spring. Spring is the normal period in Montreal, in Canada, which we'll have less here, but in there it's really a period that is important with a lot of runoff. You can easily see that above about 20 modified Fishman units, so the fluorescence units of the coliminder, you really see there are peaks above that certain threshold. And we conducted here the yellow stars, were the periods where we conducted event sampling, they were comprised of only one sample or several samples per event. I would like to focus first on this specific period here in winter spring 2017. Is a close-up of this period. We can see again the gluconase activity in orange, and below you see the river flow rate, snow cover, and rainfall, and you see these peaks of gluconase activity. The green dots are the E. coli routine monitoring data measured by the municipality, and the dark dots, the black dots, are the E. coli event-based data that we collected during those specific events along the glucuronidase graph that you can see there. So snow melt combined with rainfall, you can see here actually a snow melt with heavy rainfall, 20 millimeters of rain within one day, that triggered really sewage bypasses of nearby wastewater treatment plants. We actually, in the paper I mentioned here, we have identified a nearby an upstream wastewater treatment plant being the dominant source of fecal pollution. It was contaminating more than the nearby CSOs, and this is really causing this peak in gluconase activity. With you can see here in this brownish color the volumes that are discharged by sewage by the wastewater treatment plants, and this was triggering up to 1 log increase in E. coli during those specific events. But the question was, what about the pathogens that we detect? What is the real risk here in terms of pathogenic occurrence? So again, the same graph here, but we have added Giardia and Cryptosporidium points. You can see here the symbols for those two parasites. And with a close-up for each of the two events, you can really see that the Giardia here, specifically in the first event, was following nicely the glucuronidase activity during that event. Cryptosporidium was remaining relatively low, and during the second event, we have less points, but also it was really rising with the glucuronidase activity. I didn't plot the E. coli data here, but it was also very well correlated, as you saw on the first, on the big picture graph. So with all the data points we collected that I showed on the graph with the different stars, there were different events we actually studied, and we're combining all the data from these events plus the routine monitoring data, because that Cryptosporidium and Giardia were routinely monitored by the municipality over several years. They were measuring every year a monthly sample. If we take those routine monitoring data, the black dots, and we then do it with my colleague Emile Silvestre, a negative binomial regression model to plot these to assess the correlation between glucuronidase activity and Cryptosporidium and Giardia and E. coli. And you can see those graphs when we also added the event-based monitoring data in red triangles. And for Cryptosporidium, it was globally low. It didn't really increase a lot, less than 0.5 oocysts per liter, even during events. But for Giardia, it really increased above 20 to 30 modified Fishman units per 100 ml. You can really see that it definitely increases above a certain threshold of glucuronidase activity, except for snow melt samples, which I have identified here. They are really there is more special samples that are not really responding to the same trend. Snow melt without rainfall apparently doesn't really carry a lot of pathogenic parasites. And for E. coli, you can see here also much more data, of course, routine monitoring data, and a similar trend to Giardia. But you can really see that they are out of the relationship here, these E. coli, probably because these untreated sewage is probably discharging fresh E. coli with higher ratios of culturable versus non-culturable, which could explain then this discrepancy with the routine monitoring data that collected on the base flow. It's important to note also that these sewage treatment plants upstream are treating with UV their effluents, so that you're yielding probably also a lot of non-culturable E. coli. Whereas the CSO or the bypasses are discharging fresh E. coli that are not treated, of course. Now coming back to the map of with the river, we had the second treatment plant that I identify here in blue, and we were measuring for some period the two treatment plants in parallel with the colliminer at their intake. You can see here that there is a cumulative impact of untreated sewage release with the higher activity, globally higher activity in the downstream treatment plant in blue. But the interesting thing is that still about 5 to 10 millimeter fisherman units per 100 ml in difference increase for sewage drinking water treatment plant number 2. Interesting thing I was saying here was this peak that you don't see upstream in drinking water treatment plant 2, because this was a plant discharge of a sewage plant that you can see here on the upper right corner. Here on the upper right corner on the map, you can see the drinking water treatment plant number 2 and wastewater treatment plant upstream, only 5 kilometers upstream, the same riverbank. So there is no enough mixing with the river, so it follows really the riverbank and directly contaminating the downstream intake. And that was this peak being caused by this plant bypass, sorry. And of course, you see then also an increase in Giardia densities. Again, very little increase in Cryptosporidium, but what we also see here is kind of a confirmation from the previous case study with the previous upstream plant, is that during snowmelt, this was a larger snowmelt, as you can see here from this graph here, with little or almost no rainfall. Giardia was not necessarily high in those samples, as it was observed upstream for the other plant, whereas the glucokinase activity was increasing in response to the matrix, probably a lot of inactive E. coli or other interferences that were causing an increase in the glucokinase activity, but no increase in pathogenic organisms. Yes, here you can also see that compared to routine data, definitely those events, especially the discharge, yielded higher Giardia densities than routine monitoring data on the right-hand side. So after that, it's interesting to really detect them, but it's also very time-consuming and very tedious to target those events, and also go on-site and filter large volumes of sample and then analyze those samples. So we wanted to develop a fully automated methodology. So to design new sampling methodologies using these new online tools. So what we did is that we tried to combine an autosampler with a colliminer. So both are communicating, and a colliminer is triggering the autosampler under specific conditions above a certain threshold that we have defined preliminary using after studying the historic data of glucokinase activity. So basically, the autosampler will take a sample. We had to do some adjustments because we had to collect at least 20 liters instead of only 1 liter. So 20 liters that we would take them as one single sample, and this had to be repeated 4 to 5 times with the ISCO, whereas the ISCO normally collects 24 liters maximum. So we had here to collect 4 times 20 liters in big carboys to be able to study to detect Cryptosporidium and Giardia. So that's what we did here, and I can just briefly show you here this graph of glucokinase activity really increasing after a major CSO discharge that was in another study site, and Giardia in these gray triangles that was relatively low before and after the event, very intermittent event as you can see, but it increases really a lot as you can see here, up to almost 2 logs of increase of Giardia during that specific event. So I'll finish with some take-home messages. The automated on-site monitoring of goose activity at high frequency or near real time, let's say, enabled to characterize first hazardous events, amplitude, frequency, duration, identify main sources of fecal pollution. I didn't stress here, but in the paper we really explain how we identified the upstream sewage plant being the dominant pollution source. Demonstrate the cumulative impact of untreated sewage release on the contamination detected at the intakes. And when we look at the association between goose activity and those pathogenic protozoans, we see that Giardia levels increase up to more than 1 log after rainfall and sewage bypasses. But of course, Cryptosporidium remains globally low. And for Giardia, although it increases after rainfall, snowmelt, again, without major rainfall, appears to be less critical. And then generally speaking, here the automated approach for on-site monitoring of host activity provides a really interesting framework for pathogen sampling, be it manually or automated. And with that, I'd like to thank the entire team of at the 2 research chairs and Polytechnique Morale, and thank you for your attention.
Thank you, Jean-Baptiste. So the floor is open again for questions for this last presentation of the first session before we go into the break. I will start with one, so you have time to post your questions. I thought that I noticed that drinking water treatment plant 1 or 2, that you have a kind of daily pattern at the lower concentrations, but not at the other plants. Do you know what causes that pattern? Or do you have an explanation for that?
Yes, so the daily pattern, I didn't stress it here because I wanted to focus on pathogens, but and that's explained in the paper. The daily pattern actually helped us identify the dominant source of contamination, because when we saw this daily pattern, we were thinking what could cause it, and actually it was the daily variation in sewage, in treated effluents from the upstream wastewater treatment plant that was actually releasing the sewage upstream that was causing this daily pattern. When we zoom into this pattern, we see this M shape that is characteristic to sewage release, with an increase in the morning, then a little decrease during the day, and then an increase again in the evening, and then a decrease during night. So this M shape was really seen on the glucuronidase activity. So that was really kind of a fingerprint of an upstream sewage, treated sewage release, and that's how we could identify this as being the dominant source.
Before we have the paper, there's a question in the chat. Could you please elaborate a bit more on how Giardia and cryptos were so fast analyzed simultaneously with the glucuronidase activity?
So okay, maybe I wasn't clear enough here. Actually, crypto and Giardia were not analyzed with glucuronidase activity. Glucuronidase activity helped as a guide to target the sampling, and we sampled manually or automatically or with an auto sampler, and then these samples were brought to a laboratory, of course, to analyze Cryptosporidium and Giardia using the USP method 1623 that I showed before. I didn't go into details into the methods, but of course we don't have yet a method to analyze these pathogens on site and in near real time. That is what is expected by everyone, to be able to have a method to analyze in near real time pathogens. It will come, I guess, in years or decades, but we're not there yet. And for now, we have proxies that are enabling us to target the sampling, because as you can see on the previous graphs that these events are very intermittent, and if you do just routine sampling on a daily basis or even twice a day, we had the same case in recreational water, it's not enough. You can miss some very intermittent events, whereas if you measure at near real time in high frequency, you can really characterize your fecal pollution much more precisely and then target those specific events you want to characterize for pathogens.
Thank you. Are there any other questions from the audience that would like to be answered? A few seconds, and otherwise we go for coffee break. No, I don't see anything coming up, so let's go for coffee break. Thank you all speakers for the first sessions, for all your information, all the results. We're back at 10 to 4, so we have a 15-minute break. Hope to see you back all then. Thank you.
Thank you. All right, so welcome back, everyone. We are now, I hope you had a good break. You walked a bit. And because now we are attacking a second session that where companies, we have 5 talks. Companies will now present some case studies about their product, and starting with Jorien Favre. I'll just take your bio. Sorry. As I am the chair for this session. So Jorien Favre, she pursued her PhD on online microbial monitoring and biostability of drinking water at Ghent University in 2023. Since then, she's been working at Europe Monitoring as chief scientist, developing and researching multiband spectrophotometry as an affordable and sensitive online microbial monitoring system for clean water applications. And with this, Jorien, I will hand over to you the mouse so that you can go through your slides. All right, the floor is yours.
Okay, thank you, Jean-Baptiste. Hello, everyone. I'm not seeing my. Aha. Yes. Yeah, so thank you, Jean-Baptiste. I indeed come from a research background, but here I wanted to share some things that I noticed during my shift towards more practical applications. And I also want to introduce the orb sensor and a bit of our vision on online microbial monitoring for drinking water applications specifically then. Yes, so just to quickly start and frame, why would utilities actually invest in online microbial monitoring? I will keep this short, as I understand everybody has already a lot of background on the topic. But in short, there still remains a microbial risk in drinking water, and the current regulatory sampling takes a long time and is also giving limited insights. So especially when moving towards reuse applications, and then, for example, in the US as well, water is often over-treated, so more than is actually necessary, because we are just not sure of what is actually going on or what is actually the health risk. So yes, there is a need, and sorry. Okay. And also in Europe, the framework is changing towards a risk-based approach, and online microbial monitoring can really fill in this gap. So yeah, there is a need, and currently there are already technologies available, and this is also what I studied a bit in my PhD research. So on the one hand, we have non-health proxies. They are online measurements, but they are indirectly giving indications of the state of the water. So it's not really directly monitoring microbiology, but they are relatively cheap, so it can give an indication. Then on the other hand, there are a lot of really sensitive, informative, and yeah, just very solid online microbial monitoring technologies available. But then the drawback is that sometimes they require chemicals, and they have a relatively high investment and operational costs. So in short, there is no abundance of technologies, but when we look at drinking water utilities, they are often using these technologies in a limited way, and initial interest can stop after piloting. So why is this? Even if we get so much more information out of this, there were some of the things that I noticed was that there is no legislative value. It's also what Nikki mentioned. So it's often hard to know what is now an abnormal change, and how do we have to interpret that data or use the output. Then secondly, utilities, they are working with limited resources, and then I'm not only talking about money, but also people that are maintaining the devices or just looking at the outcome. And also the data is really informative, but yeah, as we also saw during the conference, sometimes it can get really complex to find correlations or do further analysis required. So yeah, these challenges were what I noticed when we were developing ORB, and it's also how we want to position our technology by tackling these hurdles. So we provide a sensor that you can see on top, and it's combined with a decision intelligence platform. We don't require any chemicals and are a lot cheaper and also faster than the other biomonitors that I discussed, because we work with light and no staining. We focus on the ease of deployment and the maintenance, and also try to provide assistance in the data analysis and automation on the platform together with the customers. So we really can be seen more as early warning scouts in the field, and not as a replacement of any other technology, but complementary, where we can be put at more remote areas as we don't require all the maintenance, and then other more research-based technologies can be implemented at the strategic points. And I think ideally the data can be coupled to learn as much from the water or from the data that we have. So how does it work? It's based on a fluorescence spectroscopy, so it's been around for decades already. But we have really tailored it towards drinking water applications. So we use UV light that will excite the bacteria. We are actually targeting the aromatic proteins in the bacteria or microorganisms in general. So and then they will fluoresce and emit light at certain wavelengths that we are capturing with different multibands, and we use machine learning to analyze the data, and we give you then a result in autofluorescing units. It's called the bioload of your water. So when I say practice, we are already deployed in the field. These are some of our customers, and you can see the sensor. There's a man for scale. Here we are installed in the sampling lines. And we also have several case studies published on our website, but because I see that with regards to the timing, I will quickly skip these. You can find them on the website. Send me an email if you want. We can discuss them. So it's at the production, we can detect disinfection changes. Another one is during storage, where we can understand the microbial risk. Those are available, but I'll quickly go to make my point. So just generally, there are still some challenges that are remaining. What I noticed is the cybersecurity of utilities combined with the data collection. It's always a balance between security, but also functionality. I think it's not only for us, but also for others. We want to provide good data, but for that we also need input from the customers, and there it's always a battle. Secondly, it's also not every technology, but a lot of technologies come with their own platform. It's a bit of a Wild West. I think it's also a challenge to couple all of these, and it's always a long time before we get validation or approval from our technology, as everybody wants to pilot on their own water, which I understand. And just overall, it remains a new data point, so it's always a learning curve together with the customers. I want to resign with this slide just to show you that higher resolution yeah, well, with higher resolution, you can actually start giving more meaning to your data. So it's not just about more data. So for example, this picture with 52 pixels versus 10,000 pixels, you can not only see what's going on, but you can also start interpreting it. Like you see that it's a family and you see 2 children, probably with their father on a holiday, etc., etc. So online microbial monitoring, it's a lot more than just collecting data. Thank you. I'm happy to answer any questions.
Thank you, Jorien, and thank you for keeping the time so well. It's 10 minutes, like a Swiss clock. Perfect. Thank you very much. I forgot to explain that the questions would be at the end. There is a Q&A session after this session, so please, everyone, keep your questions. I mean, you can post them anytime you want. Just maybe specify who they are addressed to. Each of the 5 speakers we will have during this session, and afterwards the Q&A session at the end, after the 5th speaker will have finished, we will address these questions and come back to the different talks. So we'll go to the next talk, which will be from Wolfgang Vogel. So Wolfgang is a mechanical engineer. Sorry, I'm sharing the wrong screen. Wolfgang is a mechanical engineer and physicist who has worked in research and development in various areas of technology. In 2010, he began developing the measurement technology used in the Coliminder. Since then, he has built up the company Vienna Water Monitoring Solutions, VWMS. He has been actively using the Coliminder devices since 2014 and has gained a good overview of the use and meaning of the enzymatic activity parameter through the approximately 700,000 measurements that have been carried out with the Coliminder devices in various applications. So Wolfgang, the floor is yours. I will just hand over the mouse, the control to you, and you can start now.
Thank you very much, especially for the organization of that interesting webinar. And yes, I completely agree with Jorien that the time resolution adds a great dimension of information to the microbial data. It was just because of the fact that the traditional method was too slow and too expensive to generate that amount of data. So we're not used to it. Although the Coliminder was already topic, I can click, but I cannot. It doesn't react.
Maybe check with your keyboard with the arrows. Maybe it will work.
So the machine has already been mentioned, or the technology has been mentioned. It's an enzymatic measurement based on an automated fluorogenic enzyme assay. Still doesn't. Oh yeah. Now I jumped in. So today I will talk on especially the use case of food and beverage industries. Just to recap, the enzymatic measurement, in contrary to the standard measurements or standard lab methods, which are based on culturability of the organisms, this is what is used to detect them or to enumerate them. So you have the CFU per 100 ml, and this is generated through proliferation. And sometimes that's mixed up with living organisms. Of course they were living, otherwise they can't proliferate, but the other way around doesn't work. It's not true that only culturable organisms are alive. And as these organisms, which are measured in the most cases, are indicators, for an indicator is unimportant if it is culturable or not. It's indicating that it's present indicates that it's there. The enzymatic measurement approach is very linear. So it's extremely linear compared to a traditional method, and it's extremely sensitive. It's well done. It's really, really sensitive. And so therefore it's a real measurement, not an estimation. Just wanted to point it out. So in food beverage applications, usually, of course, if it's about raw water, so this is a process of raw water intake, filtration, activated carbon, polishing filter, nanofiltration, and then storage tanks, and then a production, for instance, a bottling plant. In the raw water, the risk indicating enzymatic activity or the risk indicating bacteria would be E. coli, because fecal contamination would be the highest risk. But from the intake on, at least after the first filtration step or after the first filter, the risk pattern moves, and E. coli is not the best indicator for microbial risk anymore, because everything can grow on a filter. After a filter, you should switch to total activity, because anything can grow on the filter. You never know. And so throughout the process, you should monitor for total activity. And what we did here in this was a bottling plant in South Africa. We scanned through the complete system by, on one sample line, monitoring the incoming raw water to get an idea if it's fluctuating in activity. And on the second sample line, we took samples from different process steps. And so when we go through the water source, it was municipal water of Johannesburg, and then raw water tank and feed line was a little bit lower than the incoming water, because they added some hypochlorite to it. Activated carbon, filter 1 was okay, was reducing the contamination. Filter 2 was increasing contamination, means was not very good anymore. Polishing filter added additional contamination. And from the nano box, these filtration tubes here, some were good, 2 and 3, 5 was horrible. I mean, it was 4 times as much as the raw water coming out there. And here the autoclave planks in as a control. So, and all these tests have been done in parallel in the lab, standard lab method, and all of them were non-detects. And this is the reason why they had recurring problems every once in a while, had to shut down the plant, completely disinfect, because the traditional method is a lot of work, and of course, they're always looking for non-detects. So you do the work, you get non-detects, you're satisfied, you spend a lot of money and time for no information. And this is one of the great advantages of rapid methods, and especially the enzymatic one, because you can go deeper and you always get a timeline, and the timeline contains information, as Jurian said. So another use case, very short, that's one of the biggest food producers in Austria, that's RO systems, supplying the water for their production. And depending on so RO water, you would assume, okay, RO water, there's nothing there, everything's clean, but it is not the case, especially if you monitor high frequency. You see left-hand side is more higher frequency, right-hand side is lower frequency. You can assume that the pattern is the same on the right-hand side. You just don't see it because the frequency is lower. Interesting, I won't go into details, but what is this? This is a weekend. What is this? This is a weekend, because there's less demand, and therefore overall activity goes up. We have several of these installations, several customers here, Romaqua monitoring the carbonated, naturally carbonated, and plain mineral water, so the wells and the final products. This is a bottling food beverage company, a big one, and in Germany, they have already switched a lot of their lab protocols from CFU to enzymatic activity. They just determined by parallel testing and then setting their limits. So I lost my, here. So it really works in practice. They save a lot of lab waste. They can do more frequent measurements on their critical control points. They gain in safety. And in order to, because when you see here, when you see here, we have this 10-fold sampler allowing to really efficiently measure grab samples, but you have all the silicone tubes, and that's not really ideal because measurement is very sensitive, and if you don't clean them well, then you have contamination. So we developed a sampling carousel. It can be applied with a 12 sample dish and a 24 sample dish, and then you can automatically have these samples measured. You can even seal it with a parafilm, and the needle will go through the parafilm and take the sample from these individual samples. So as we have built it up and I had a lot of fun because it works well, we went to the shop and purchased 12 different mineral waters, still mineral waters, and put them in and had them run three rounds in the carousel. And this were these different waters. So first round, second round, third round, six hours, one complete turnaround if you do full cleaning in between. The advantage is there's only one sample line and that's cleaned every time. So, but this is a very interesting thing. We could stay with that, I think, for hours. I have 14 seconds left. There's difference in the microbiome, and there's difference therefore in the measured activity. There are some that are developing, that some are constant, means stable microbiome under air influence. There's some who are decreasing and some who are growing. I can just kind of guess that might be the microbiome, some more aerobic or anaerobic, and if they then see the oxygen, then they reduce, and others like, oh, so 10 minutes. Yeah, so that was very interesting. Sorry that we can't go longer. We have installations in pharma as well, because we can go down very low. That's also interesting, but that would exceed the limit. Thank you very much.
Thank you, Wolfgang, for also keeping the time. Very interesting talk also as well. We have a really interesting session here, session number 2. We have 3 other talks. Again, if you have questions, keep them for the Q&A session. I have already some, so it will be, I think, an interesting discussion also. So next talk will be, let me see, I will just change PowerPoint. So the next talk will be from Sylvain Kaufmann from Binovate. I will just hand over the mouse and so Sylvain Kaufmann is a solutions program manager at Binovate, where he supports water professionals in tackling microbiological challenges with innovative products and applications. Holding a degree in biomedical engineering, Sylvain began his career in medical diagnostics and now worked for over 10 years in the development of novel flow cytometry solutions for agtech, biotech, and water. In his free time, Sylvain is a fresh air enthusiast who enjoys all kinds of outdoor sports, especially those involving water in any of its physical forms. And I guess in Switzerland, you have many things to do outdoor. So Sylvain, the floor is yours for 10 minutes.
Thank you very much. Did you hand over the presentation already?
Yep.
Yeah, perfect. Just quickly check whether I can skip slides. Yeah, that works. So thank you very much for the nice introduction. Good afternoon, everybody. It's been a very inspiring event so far for me, and I would like to congratulate to everyone, all the researchers and also sensor companies for your commitment to innovation and for sharing your achievements today with everyone. It was a very, very nice journey so far. And as Jean-Baptiste mentioned, I'm representing Binovate here today. It's one of the sensor companies. Probably most of you have heard of us already. And I have brought with me an interesting case study on the use of flow cytometry in water reuse systems. And we will have a closer look at membrane filtration. So I would like to talk quickly about the Binovate. We are an SME, approximately 50 people, founded in 2011 in Lausanne, where we still have our headquarters. We're commercially active in Europe, North America, and Asia. And most know us from our industrial flow cytometric solutions, what we call BactoSense technology. And as you can see here on the right, our solutions are already used by quite a number of different companies in different industries. So over the last couple of years, we've established quite a strong foothold in water utilities, but more recently also moved into industrial waters like cooling systems, food and bev, research, and most recently into pharma, personal care, and medical. But today, I would like to talk about membranes. And for those of you who know The Lord of the Rings, probably know Gandalf's famous statement, "You shall not pass." And that's exactly what membranes do. They don't let certain things pass through. And membranes are really at the heart of so many different processes. I just try to collect here a number of them for you. For instance, we have membrane processes in the production of purified water for pharma water systems. We use them in water reuse systems. We treat blown down water in cooling systems. We use them in desalination and so on. So they're widely used. They're very powerful methods or techniques, but they're also not invincible. And we all know that microbiology can be a challenge if you work with membranes. We know that microbiology can affect their performance and eventually even have an impact on the water quality. And keywords here are biofouling, regrowth, and membrane breach. So on the one hand, we have these microbiological pain points or risk factors, and on the other hand, we have membrane integrity test methods that cannot really measure microbiology. So that puts operators into a kind of a difficult spot. And that's why people started to look for complementary solutions to basically address those microbiological challenges. And microbial sensors is definitely something that can complement any kind of operations of membranes and address these kind of pain points. We've heard a lot about flow cytometry already today, so I won't really go into the details. More important is that there's been quite a number of scientific studies lately that propose the use of flow cytometry to address those microbiological challenges. There's papers that propose the use on direct integrity monitoring. So you install an instrument on permeates or filtrates, then detection of biofouling, and looking at the microbial regrowth in permeates. So how could that look like in practice? And now I come to my case study. So I would like to show you that with a recent example that we had with a pharma company. The pharma company is producing a biopharmaceutical and has one production facility in Switzerland. And their target is to reduce water consumption. They have very, very ambitious sustainability targets. And water reuse is one of the avenues that they would like to explore. And the reclaimed water would have to meet Swiss drinking water standards. And one of the challenges that we faced in that trial was that the feed water that goes into that water treatment system has a very big variability on its composition. So over time, that composition can really fluctuate a lot. And particular attention had to be paid to give that customer a lot of confidence in also in the microbiological quality of that water that comes out of that treatment system, because that was one of the really important KPIs. And looking for solutions, they came across EcoPack. For those who don't know EcoPack, it's a Belgian company. It's a leader in sustainable water management solutions. And EcoPack then deployed a state-of-the-art water reuse treatment train, containerized solution, as you can see here on that image, and basically conducted that kind of pilot study. And we included the BactoSense into that because of that important KPI, that confidence in microbiology that customer required. So now let's have a quick look inside. So we have the treatment train depicted here. We have a coagulation step and then UF, and then we essentially have just a two-stage RO system. So water goes into a tank through a pre-filter and then into RO1, and then from there concentrate is being fed into the RO2 path, again through a tank pre-filter and through the second RO unit. And what we mostly do if we come across a system like this is we start by taking grab samples from critical points along the treatment train just to get kind of a global picture of the microbiological landscape. And that's what we did in a kind of a first step on the first day, just taking grab samples all along the treatment train. And what we found is that we go into the train with roughly 45,000 cells, so that's a total cell count, and we have a first significant reduction across the ultrafiltration membrane, so it's a 1.4 log reduction that we detected here. And the second big reductions are the RO1 and RO2, 1.6 and 1.7 log reductions. Respectively. What was even more interesting was that we had kind of a significant regrowth of cells in this part here, so water tank and pre-filter. That's something that we would not have expected to that extent. So for instance, in that case here, we have cell counts, 1,600 total cell counts post UF, and then after the water passed through the tank and the pre-filter, we were shooting up to almost 25,000. Even a bit more extreme was on that leg. So in the end, we entered the RO2 system with more than 200,000 cells per ml, and that's something that was a bit of a concern. So we had a closer look at these pre-filters, identified them to be contaminated, swapped them, and brought those cell counts significantly down. So kind of a first action could already be triggered after just a day of sampling. Then we also had online monitoring installations. So one BactoSense that was moving from point to point. So kind of an at-line installation, sampling every 60 minutes, or also another time period, depending a bit on the experiment. And what we wanted to understand is the effect of process variations. So in that case, the membrane recovery, when we kind of changed the feed pressure. Wanted to understand what kind of effect that has on microbiology, and that was a big surprise. So we saw a more than 20-fold increase in the total cell count when we changed the membrane recovery by increasing the feed pressure by just 20%. So that was really a big surprise to us. That was clearly not expected, and that's already my first take-home message for you. If you do process optimization, don't forget about microbiology. It sometimes behaves in ways that you may not expect. Don't really have the time to go into all the details. I just summarize here the key insights from that trial from our perspective. If you monitor membranes on filtrates or permeate site with your sensor, go as close as possible to the outlet of the membrane. Otherwise, you might capture some other things like microbial regrowth in the pipe system. Then UF filtrates and RO permeates are definitely not sterile and also not biostable. I would like to make the link to what Wolfgang already presented previously. It's kind of a misconception that some plant operators sometimes have. So there's always cells in there. Then we also saw some effects of stagnation. So short-term stagnation really led to spikes in the total cell counts, and long-term stagnation even had kind of long-lasting effects on the water quality. Then we've seen the example of RO pre-filters that can release relatively high bacterial levels. So don't just focus on your membrane itself, also look at treatment elements next to the membrane, pre-filters, tanks, things like that. Don't forget about these. And then maybe to come to an end soon, if you do process optimization, as I said, look at microbiology, don't forget about that. So to summarize, microbial sensors can help you to better understand your process and enhance membrane operations. Most sensor types give you kind of a dual operation mode. You can do grab samples and you can do online monitoring. Grab samples help you to get a very big, quick overview of a system. You can identify critical control points or hotspots. and also allows you to look at individual modules. If you have a big membrane rack, many modules, you can kind of identify which module is failing. That's a spatial dimension. If you want to look at the temporal dimension, you obviously want to apply online monitoring. This really helps you to capture microbial dynamics. It's very useful for process optimization. And also some of our customers are using membrane integrity monitor or BactoSense as a membrane integrity monitoring tool, especially after commissioning of new membrane systems to detect any kind of burn-in failures over the first couple of weeks of operation. If you want to know more about what I just told you, we have a nice application note on our website, or you can scan the QR code here, and it gives you kind of the whole application landscape in membranes. But then also we have a case study, the one that I just presented, with much more data than I could present here. Feel free to reach out to me directly if you have more questions, or subscribe to our newsletter on our website. Yeah, that's it. Thank you very much.
Thank you very much, Sylvain, for this interesting presentation too. So again, we'll keep questions, and we will now go to the next flash presentation. Will be presented by Job Appels, and I will just briefly introduce Job. So Job Appels is the managing director of Microlan, a Dutch company specializing in real-time water quality monitoring solutions. With over 3 decades of experience in environmental biotechnology, Job has been instrumental in developing innovative systems like back control. You've heard of it during Nikki's talk, which enables continuous online detection of bacterial contaminants such as E. coli in surface waters. His work has significantly contributed to public health initiatives by providing early warning systems for bacterial contamination, notably implemented in cities like Breda in the Netherlands. And Job's commitment to advancing water safety technologies has positioned Microlan as a leader in the field of microbiological water monitoring. So Job, the floor is yours.
Thanks, Jean-Baptiste. If you give me access to...
Yeah, you have access now.
See if it works. Yep. Okay, thanks for this introduction. Kindly collaborated with ChatGTP on my LinkedIn page, so too many positive words, I would say. I'll give you a short introduction because I realized I have too many slides. A lot of information has been passed already by previous presenters. I'll focus on the river and bathing water monitoring, specifically the last issue, but there are many things already mentioned before, which we do about in the same way, but we focus on some references. As I introduce you a bit about the company, we have 3 technologies. We'll focus on the bacteria monitoring. All of them give you the possibility, also the different instrumentations which have been shown by previous presenters for high frequency data but low in quality. But it gives you an idea about trend monitoring, and as also been told by previous presenters, you can be more in control of your water. That's the main use of online microbial monitoring. One slide about the company, started in 2003. We've come up now with a new instrument, the Back Control 3. Nikki mentioned before in the previous test by KWR that one of our systems was not so sensitive, but that was actually not the newest system we have, which we are now going to introduce quite soon. We'll focus on the 2 different topics, mainly the monitoring of microbial loads of rivers, because they're used quite a lot for drinking water production, and the Netherlands is heavily related for at least 1/3 of the drinking water production on the river water quality, but also on the bathing water issue. As mentioned in previous presentations, also from Coliminder, from Wolfgang, the traditional methods compared to the enzymatic monitoring, the biggest advantage is that you can have a quick result. And this has actually been previously developed in back in 2000 and presented by Professor Farnleiter from Vienna University. Same like with other presenters, we are focusing really on steps like we do online monitoring, and you have to take a sample when it's really important. So you see, on the left side, you see a peak. That's an intake company. You want to take a sample at that moment and see what the causes of the contamination, or a slow increase of a baseline. On the left side, that's actually E. coli monitoring. On the right side, it's total bacteria. But in both cases, you know when to take a sample is at the moment when there's a baseline change. And then this can always be followed by by lab analysis. We do more and more also combinations with DNA analysis like qPCR on next generation sequencing, which I will also present a little bit more down the line from this presentation. Correlations of the 4 technologies we can do are shown here. I won't go too much into detail. These can also be found on various international publications. Short difference with other providers is that we have a pre-concentration step inside. So we have a 0.45 micron filter that will capture, of course, the E. coli bacteria, which are bigger. You add a buffer, you add, and then the reagent is causing that the beta-glucuronidase gas is splitting off, and a fluorescent compound, methylbariferon, is produced, which we can measure. And this is a continuous process which gives you, as I mentioned, a result every less than 1 hour. This has been published also by KWR to make, for the bathing water, a guideline when to set up an alarm. And of course, 1800 MPN is the European guideline for that. And if you do, it's quite difficult to do a real correlation, but if you take dilutions of wastewater or sewage water, which is going into the specific bathing water area, then you can make a nice correlation, and you can see when you have to set your alarm, but it's really depending on the location, of course. To give you a few examples, this is actually quite an old one, so we're not talking about new technology. I think this is already like maybe 10 years ago. This was done in Denmark, seawater application. Children were actually getting sick when they were playing in the pond on the right side of the top picture. So what the local government was doing, putting the instrument into a mobile system, put it upstream, and you can see there are many overflows or many alarm levels in the red part of the diagram. And at that specific moment, the sample is taken, and then qPCR was actually used to track down where the pollution was coming from, if it's human or cattle, for example. So you can trace where the pollution is coming from. And this is a very good example on which we have based our future developments. Currently, we're doing quite a big project in the city of Breda in the south of the Netherlands. They actually approached us because they had several bathing events, like people swimming in the canals, which are not actual swimming water, bathing water. And on the bottom part, you can see quite a big event with a lot of students being in the water with a nice festival. But we have developed and collaborated very strongly with the local city government and the water authorities. And after a 3-year study, we have come up together with our partners with some very interesting results. Actually, the government chose to show on the website to warn the public if the water was really going from orange to red. Orange meaning, yes, you can do like supping or something like that, but don't swim in the water. And if it's red after an overflow, after an alarm from our instrument, it means, well, it's very dangerous to go in the water because it's actually, or on the water, it's actually sewage water. And this is a public information which the government really wants to inform the public about the status of the water. There was quite a lot of research being done, which is published. You can see the blue line is from the bug control, and there's a strong correlation with the overflow and also the rainfall. Of course, in a previous presentation, it was mentioned 13 millimeter for a day, but what we see, this is also, of course, a different location. If you have between 10 and 50 millimeter of rain, especially heavy rain and peak rains, which you see more often due to climate change, an overflow happens, and then within 6 to 8 hours, we see an alarm. Automatically, a sample is also taken, and somebody from the lab is sent to take another sample and to show with qPCR if the water is still safe or unsafe for the public. This has been documented in a practical publication. You can find it on our website. From the first pilot phase, we switched on to a data as a service project, which we're now in the 3rd year, and the government is really happy with that. And then we switched to some other applications. We do quite a lot now in river monitoring. This is at the local location IJsde, which is close to Maastricht in the south of the Netherlands, and the river Meuse is used for about 3 million people for drinking water production, and the quality is getting worse. So the government wanted to find out if there's changes in the water microbiome, and this is related, of course, with the total bacteria monitoring we do. And the moment we see an increase, we see a high level, an automatic sample is taken, and then with NGS, the water biome is actually analyzed. This will be published later. There's quite some details inside of this picture, but it's too short to go into depth right now. Same is done on another location, also with an automatic sampling unit. So the last slide I wanted to point out is that there's coming up water swimmable cities in June in Rotterdam. I know Polyminder is also going to be there, and several cities will be visiting this, and there will be a presentation also about the same topic, same as the water micro, where Nikki van Bel is also doing a poster presentation where we'll also be available. So with that, I hope I'm within the time, and I come up with the conclusion that, as previous presenters has been said already, online monitoring is really a key help to improve the water security and the water quality. So I hope we have enough attendees. We came back from 65 to 44 attendees, that we can have some questions later on. So thanks for this.
Thank you very much, Joop, for this presentation. Very interesting too. A lot of things to discuss.
Yes.
Well, now we're coming to we are a bit late because actually you have been very good in keeping the time, but actually there is also a bit of time in transitions between the presentations. That's why we are a little bit late, but it's still okay. And we are coming to the last presentation, sorry, from Sophie Hen. Oops. I will briefly introduce Sophie. So Sophie Hen is currently Customer Applications Director at Redberry. After 17 years as engineer at Eau de Paris in France, working on microbiological sensors for water quality and involved in the Paris 2024 Olympic Games for the monitoring of the Seine quality. She joined the Redberry Society, providing solid phase cytometry devices end of 2024. She is now responsible for managing and optimizing the applications used by clients, ensuring that they effectively meet their needs and expectations. And today, she is going to present us a comparison of two rapid methods for total flora enumeration in water treatment plants. Sophie, the floor is yours.
Okay, thank you very much, Jean-Baptiste, for the invitation. So I will show results obtained by one of our customer working. It's a tap water producer in the US, and he's producing. I don't know if you okay, sorry, I was. So, and he's producing tap water using raw water from the Michigan Lake So the objective of this study was, for him, the auto control of the water quality at all stages of the water treatment train. And for him, what was important is the direct count of total viable cells, including viable but non-culturable cells, and time to result lower than ideally 10 minutes. And he did on-site assessment during 3 months using assays performed on the plant lab by his team in charge of the water controls. So he compared 2 methods. The first one is the online ATPmetry here by Hack. I think everybody knows how ATPmetry online works. But so the ATP is the energy contained in the cells. So after an extraction, the ATP will do light with an enzyme with luciferase. And it means 1 bacteria contains 1 femtogram of ATP. And the other method, alternative method, is solid phase cytometry. Just try to okay, so it's solid phase cytometry. That was the red one by Redberry. So how solid phase cytometry works? So the sample is filtered on the 0.45 membrane, and all the cells will be sticked on the membrane. So all the rest of the background will go through the membrane. You just have the cells on the membrane, and all the cells will be stained with a staining agent, which will look after esterase. So during the time to result is for 10 minutes. So each minute the device will make a photo, and you will have an increase of the fluorescence in the cell. For here the 5 you have here 5 photos from E. coli, for example, and you see during the time you will have an increase of the fluorescence. Whereas when you are just having a look at inner particle, the fluorescence will remain the same during the 10 minutes. So the kinetic is flat. So, for example, for E. coli, you will have an increase of the fluorescence. For other type of bacteria, for example, Pseudomonas, that has efflux pumps, the fluorescence will get in and get out. But the algorithm can make the difference between something living and an inert particle, and that in 10 minutes. So that was where the two techniques. So the first part of the study plan was the determination of the limit of detection of both techniques. So we filtered, we did what they called negative. So we filtered water at 0.2 micrometer, and we did we test on red one, ATP, and R2A plates. So for the solid phase cytometry, we have a limit of detection of 10 cells, whereas for the ATP, we have a limit of detection of 1,000 cells for negative controls. After that, they had a look on, they did an evaluation along the water production train. So during, you have here the ATP results, and the other side, the cytometry, the solid phase cytometry results. So you can see that we have a strong repeatability in both countings, but we have higher counting with ATP than solid phase cytometry, sorry, on all waters, except here for filter influent. So if we go more in details, For the raw water, we have in mean for ATP, we have 4,000 cells, whereas for the red one, we have nearly 5,000 cells. So we can see that we have a strong correlation between solid phase cytometry and R2A, and a very lower correlation between ATP and R2A. And we have here a low counting with the red one. The question is maybe why? So this can be because of the lysis step. For ATP, we not only lyse bacteria, but algae and protozoa too. For the focus on the filter influent, here we have another difference. We have lower countings with ATP, whereas we have a higher counting with red one. So when we had a look on the photo on the red one, we can see that we had a biofilm which was visible on solid phase cytometry. And here you have the counting that I said, so 30,000 cells measured by ATP and 21,000 by red one. So here we have a biofilm visible only with the solid phase cytometry. So that's why we have a higher counts on the solid phase cytometry. And here you can see the photos. So each dot here in the biofilm will be counted by solid phase cytometry, whereas on the plate here we just have one biofilm, one CFU. That's why the difference between both techniques, and we can expect that the lysis buffer cannot extract all the ATP from the biofilm. And here the last one, it's the plant effluent, so the water produced. When we have a look on the ATP, the ATP doesn't count below 100,000 cells, whereas the solid phase cytometry, we have, for example, a mean of 9 viable cells, but in ATP we have a little bit less than 3,000 cells. So we have a strong correlation between solid phase cytometry and R2A, and much higher count on ATP. So that's not relevant for monitoring at the end when you have very low counting. So the conclusion of this preliminary study, auto control of water quality and treatment efficiency is visible in real time. Here we have a time to result of 10 minutes with solid phase cytometry on all types of water along all the production train. The limit of detection is of 10 cells per ml, and we have for ATP the limit of detection is 1,000 cells. We have a strong correlation between plating on R2A and solid phase cytometry, and a very low correlation with ATP versus R2A. And an additional added value for a solid phase is the revelation of biofilm presence that will be counted on plates, and the lysis buffer cannot extract all the ATP from the cells contained in the biofilm. So thank you very much for your attention.
Thank you very much, Sophie, for this interesting presentation. And now we have arrived at the Q&A session. So I would now, Nikki, do we have some questions in the chat? If anyone has got questions, otherwise we probably have some questions too here. Do you want to start with one or the other questions?
There's no question.
No, I don't see any
No.
question for now. Do you want to start?
Yeah, I can start with the question for Silvan for the Binovate. You showed the use of the Binovate, the BactoSense for the membrane removal of bacteria. You showed 2-log removal, maybe 1.5 to 2-log removal. Do you know what you can show the max? I know it's dependent on what goes in and what comes out, especially what comes in. But what kind of log removal can you show? Because that's, at least for drinking water production, that's very important. I can assume for reuse of water in the soda factory, it's also important, but they don't need 5, 6, 7 log removal, but the drinking water company would like to have that. Can you give some insight in that?
I think as you correctly pointed out, it really depends on what your feed is and how well your membrane performs and what kind of membrane system you're working with, whether it's a microfiltration, UF, NF, RO. Most of the customers that we've been in touch with have membranes that perform in that kind of order of magnitude, 1 to, let's say, maybe 3 log reduction. You find examples in literature where people have demonstrated up to 4 log reduction detection using flow cytometry. That's definitely feasible. I don't think it's really the technical limitation. If you go up to 4 log reduction, it's really the performance of the system that determines that result. Does that answer your question?
You mean the performance of the BactoSense or the flow cytometry, or how the membrane, how well the membrane is?
How the membrane, yeah, the performance of the membrane itself, and how close you actually come to the output of the membrane module. We have seen, as a side note, from the data that I presented, couldn't really present it, but we've did some kind of regrowth study post membrane, and we just had some sampling points available a few meters down the line towards the final point of use, and you could basically see almost every meter adds some microbial, contributes to the cell counter. So there's kind of bacteria that are being released from the biofilm meter by meter. So that really has an effect if you want to do an accurate crediting of a log reduction value. Go really as close as possible to the module itself. I don't really think it's a technical limitation of the sensor. It's the performance of the membrane, because as I've shown, we had a post UF, we had cell counts TCC of 1,600. Post RO, we went down to sometimes in the few hundreds of cells. We have been working with partners, BWT, working in the pharma space. They measure zeros, intact cell counts in WFI or purified water systems. So BactoSense instruments can definitely measure down really to very, very low levels. But then we also have projects in surface water applications where we measure in the millions. So the dynamic range can be relatively big. So that's why I believe it's really the actual performance of the membrane. It's not a technical limitation of the analytical instrument itself.
Thank you.
So we have one question here for Wolfgang Vogel talk on Coliminder. So the question is, the last experiment showed increasing or decreasing counts depending on the water brand after time. So that was the different mineral waters. I find this highly interesting and would like to know if this is more than one replication for the different time slots. Maybe it was just by chance? Question mark.
Yeah. Short answer, no, was not by chance. It's repeatable, and it's a property of the different waters, which is another step of information or discussion. The water has a microbiome, and there are different microbiomes, and they behave differently. And it's not only it's not. I think you should not charge or determine the quality just by one parameter like microbial activity or heterotrophic plate count, whatever, because these mineral waters have a maybe healthy, positive microbiome, and you have to evaluate that in more detail. If you apply a rapid method in a water like this, it's not about the absolute value, but it is about the time, the development of the microbial activity or parameter over time. It is always what we suggest to our customers is monitor your water, do your standard tests, and then you will get an idea of what's going on over time and what is your baseline. What is what you want to see? If the baseline goes up, it means something's proliferating in there. The microbiome is changing and it's getting more intense. If it goes down, then something is poisoning your microbiome or harming your microbiome. That might also be an indication for something's not okay. And in this case, yes, it was repeated. The test was repeated. It's actually, so we bought same brands, new bottles, did the same, got the same effect and the same results. Regarding the absolute values and regarding the development.
Okay. Thank you,
Maybe
Wolfgang.
one more comment to Silvan's answer. This is a very important point. You said you shouldn't be close to the membrane in order to avoid any contamination. This is something that applies, of course, in every method, but the machine, the device always measures what you provide to it. And not what you want it to measure. You need to make sure that what reaches the instrument is representing what you want to know about, and that it doesn't change on the way to the instrument or during the transport if it's in a tube or a car. This is something we always have to emphasize with our customers because sometimes it's not recognized.
Absolutely. We have a question for the last presentation for Sophie for Redberry. How is it possible that ATP measures more than 2,000 cells in the finished drinking water, whereas the red one only detected 9 cells? Thank you so much for elaborating. Maybe I can come back to the slide. It will be probably easier to visualize. I can hand over to you again.
Thank you. Using a solid phase cytometry, you don't have any background. So all the rest of the water will go through the membrane. You will just have the cells on the membrane. When you use ATP, you can lyse other things than bacteria, and you have always a background noise. And this background noise is not present after filtration on solid phase cytometry.
Okay, thank you. I hope you convinced someone who asked the question. It is short and clear. One question from Job for everyone. Can anyone share experiences on pre-filtration possibilities? So anyone can reply.
Pre-filtration what?
Yeah, maybe Job, you can maybe elaborate on your question, please.
Yeah, I mean external pre-filtration, if you do like sewage after disinfection or surface water, for example, with heavy turbidity loads.
Yeah, well, shouldn't be. I mean, you need to correct. If you do an optical measurement in a sample that has, if you do an optical measurement in a real world water sample, you need to correct for turbidity. Otherwise, you get the results are nonsense. I mean, at least if turbidity is variating. So, and I think that's yeah, just logical. But I think
I know. I was hoping more for maybe from the attendees' experiences or questions.
Well, if anyone has got inputs for Job, you can also contact him quite easily, I think, by email. Here we don't have any anything in the chat, but yeah, maybe someone has got a clue here and can contact you then directly.
Maybe I can add something from Binovate's perspective.
Yeah.
We don't do a lot of surface water monitoring, and I guess for this kind of application, that would be a very relevant accessory. But we do, whenever we do any kind of abstraction from surface water systems, recommend to use a pre-filtration unit. And I cannot really name you the specific models that we use because I simply don't know. But if you want to reach out to me, I'm very happy to maybe share our experiences with you. Yeah.
And in case, in case of biological measurements, I'm always a little bit skeptical about pre-filtration because microbes love to stick to particles, and you always take away something. So I'm a little bit skeptical about the meaning of microbial measurements if you pre-filtrate. I mean, it depends what size may be. Of course, you can filtrate away stones, like big ones, but you should always evaluate if you don't not take away anything of what you want to measure.
I agree, taking away and adding. So
yeah, adding as well, because in any filter is an ideal growth bed. So this, you have to be careful with that. Yes.
Yeah.
Yeah.
Another
So
question
of
here
course, combination
from
attendee. For example, with ammonia.
Oh, sorry. Yep, go ahead.
Jean-Baptiste.
No, go
it's
ahead,
a combination, of course. Go ahead. We have questions like this also with ammonia analysis, for example, and TUC. So you see it also quite relevant with more chemical analysis.
We have another question from the audience. Has any innovation traced the impact of animals, example, chironomids, on microbial activity in disinfection of surface water? So very specific question. Anyone wants to reply on that?
What animals?
Chironomids.
They're type of mosquitoes.
Yeah.
Google just told me.
Yeah.
Non-biting midges.
Surface water, disinfection of surface water.
Effect. I know when you take a glass of water, you take a fly and drop them in, then you see an effect. If this is effect, then yes. But on a real surface water, I don't know. I don't know. How many of these insects?
Yeah, probably eDNA or so. You might be more specific and trace back some species like this, but I guess all the other methods are less specific. And what we've been discussing today are less specific to these kind of animals. I think the best method would be eDNA, like is being developed more and more now. That's my.
Of course, it also depends on the parameters. If you want to evaluate the effect on, like, the animal or the fly on the sample, then you need clean sample, put the fly in and look what is changed in the measurement. Yeah,
because flies carry also fecal bacteria, but I think here it's more tracing back the presence of those specific animals, I think. So in that case would be a more specific method. I don't have any other questions in the chat. Maybe you have,
Yeah, we have this maybe from this question regarding the UV. So because
yes, indeed, there was a question from session one. Go ahead, Wolfgang, and then I think that would be the last question of the day.
Yeah, because I was thinking about, so UV is a treatment that is used widely in all kinds of waters and always and very prominent. And I can't think of a method, a rapid method, which would be able to immediately measure the effect of UV like the culture-based method does, as the UV is destroying the DNA and those organisms are then not cultural anymore, but they're still alive. Therefore, I don't know, maybe there is a method or possibility to directly measure the effect as it is showing up in a culture-based lab method. The other question is, what does it mean? But any idea how a rapid method would be able to determine this change that is caused by UVing our indicator organisms? Let's put it that way.
Any insights here from participants?
I think obviously we from Binovate, we would like to make a proposition to that question. Most of you were probably aware that we have launched a new reagent cartridge last fall. And this is kind of a complement to our existing offering of total cell count and intact cell count. What we now offer is the so-called active cell count cartridge. And the main application field we see generally in disinfection studies and also particularly with UV. And essentially the main motivators was we had kind of a shortcoming on UV because membrane stains or intercalating dyes cannot detect the effect of the UV treatment. So they're kind of membrane integrity methods, and we developed for that an esterase-based assay, and if I'm not mistaken, that's the same type of assay that also the Redberry system uses. So it's a CFDA as a substrate. It's being cleaved by this ubiquitous enzyme esterases into a fluorescent product. And I can now share some empirical data that we have retrieved in the development and validation of that product. So what we have found is that especially medium pressure UV systems, they're kind of broadband, they don't have a single wavelength, so they don't act only on DNA, they can also have effects on proteins, on enzymes. So what we have seen that we have very rapid effects, treatment effects visible on this esterase activity, especially if you work with this medium pressure UV system. So our hypothesis of the mechanism of action is not really that it's affecting the DNA on that kind of fast kinetics, that it's more affecting protein 3D structure using either denaturing proteins directly or via reactive oxygen species. So what I can say from an empirical perspective, we see very quick effects upon the medium pressure UV treatment on this esterase activity, and that allows us to then see cell count reduction. So we don't do bulk measurements, we do single cell enumeration. So we see a decrease in what we call the highly active cell counts. These are cells that have a high enzymatic activity of these esterases. They give a bright fluorescent signal, and when you subject such signals to a medium pressure UV system, you see immediate reduction of those cell counts, and that's why our value proposition.
What is the time span? The time span. Is it
we have a time to result of 45 minutes, and I think
okay.
that's 45
minutes. Is, yeah, 45
and
minutes.
Think,
okay. This is the issue. We see after 45 minutes, we see it as well. But you know, after 45 minutes, if it's a discharge of a sewage plant, it's already inside the river.
Okay.
This is so in the practical use. This is the use case is I have a sewage plant. I use UV disinfection. I want to verify that my sample or my treatment is fulfilling the regulations and that I'm compliant. And then we need something really immediate. 15 minutes usually reaction time, whatever, and then is the river, and then. But the very interesting, and what the amount, the percentage of reduction, because it will after 45 minutes not go away completely. It will take longer to really decay, I assume.
That really depends now on the microbial composition of the water. I mean, if you look at UV inactivation studies of different microorganisms, they behave very different. I think they have really log-fold differences in the inactivation rate. So I can say that the water type really has a big effect. And also, I guess the sampling location, if you would sample further downstream of your UV treatment, you would probably have already a bigger inactivation rate because it's I mean, yeah, you need to fully understand the kinetics of the whole inactivation process. And there must be an effect directly on protein level. I don't think if it was purely affecting the DNA, we would see these effects so quickly. I think if you look at DNA, transcription, translation, all this process would take some time. So there must be secondary mechanistic effects directly acting on protein level. And that's what we explore with that cartridge.
This is the great thing about the rapid methods, that we get so many new questions and interesting things to think about, and to maybe develop experiments and do research and learn how complex these systems are.
Yeah, and I think that's a very good way to wrap up this webinar. And I think indeed we are progressing very much with these rapid methods and high frequency methods, discovering new things, putting new Googles on, and seeing other things like the processes in high temporal frequency. But new questions arise, of course, like all the time in research, and that gives us some work to do. Today we've really traveling through different methods: FCF, flow cytometry, ATP, PhysChem, solid phase cytometry, enzymatics. Everything has been more or less covered today in this webinar. We're also been through surface water, karst water, going through drinking water treatment plants, up to the network, and even bottled food industry. So it was really interesting. Thanks, Elaine, everyone to accept to present during this webinar. I hope our attendees also had lots of information and learned things and have new questions that have arisen. And we are looking forward to a next edition that will have to be determined, and hopefully the platform will be fully operational soon, so that we can keep you updated. We have now a mailing list. We are part of the community of practice, and of course we will keep you informed about the new developments of this platform to make it fully operational, and everyone can also participate and to this community of practice. So with that, I would like to thank Nikki for chairing the first session, and a special thanks to Christelle, who here at least organized and made everything run smoothly for the webinar. And again, thanks to everyone for attending the webinar. Hopefully see you soon for the next edition. Bye.
Bye-bye.
Bye-bye.